I'm Dr. David Shields, the chairman of the Carolina Gold Rice Foundation, and it's my distinct honor today to introduce our guest speaker, Dr. Anna McClung. For those of you who don't know Anna, Dr. McClung is the director of the Dale Bumpers National Rice Research Center in Stuttgart, Arkansas, and is the research leader in rice production and genetics for the USDA. She has been in this position since 2005. The decade between 2005 and 2015 has been a decade of extraordinary developments. Indeed, a decade the likes of which have never been seen in rice cultivation in the United States. It was during this period that the rice industries in California and Texas became imperiled by the diminution of water resources, and it is during this period that scientists became aware of rice's penchant for concentrating naturally occurring and artificially deposited arsenic in the soil. And it's during the past 10 years that the United States has been struggling with a growing international perception that the quality of commercially produced rice in the United States is declining. We've also seen the riptides of politics and commerce alter the control of seed in the United States, the proportion of public versus private proprietary research in agriculture, and the level of funding for governmental efforts to understand and police the nation's food crops. Also, during the past 10 years, the scientific tools for understanding the genetic codes influencing desirable phenotypes in rice have become increasingly advanced. To say the least, the past decade has been the great era of challenges and opportunities in the field of rice cultivation. And we've been extraordinarily fortunate to have someone in the director's chair with such astute judgment, such skill as a researcher, and such goodwill toward those who wish to preserve the best in rice agriculture and to improve the quality and character of the strains of rice being grown in the United States. Dr. McClung is a long-standing friend and close ally of the Carolina Gold Rice Foundation. Before she became director in Stuttgart, she oversaw the genetic focusing of the old landrace Carolina Gold into the Carolina Gold Select that we use as seed rice today. And she did much of the field work, refining Charleston Gold, the aromatic offspring of Carolina Gold, into a distinct strain. Even in the midst of her ongoing efforts to determine the genomic signatures for crop resilience in the face of climate change, in the midst of her ongoing projects in rice breeding, and in the midst of her explorations of the various biotic and abiotic stresses governing crop loss, Anna McClung has retained a keen interest in Carolina Gold. And today, she comes before us in order to make a very exciting proposal. Will you join me in welcoming our guest speaker for the spring 2015 meeting, Dr. Anna McClung?
Well, good afternoon, everybody. It's a pleasure to be here, and I hope I can live up to that introduction. I've had a wonderful ride with the Carolina Gold Rice Foundation, and it's been exciting to me to be able to work with these people and to share in your enthusiasm for rice And I'm just very excited to be part of the group here and to participate with you. Today I want to share with you some of the work that we're doing at our center and some new ideas that we've been able to develop as a result of some of our explorations into genomics of rice. And there's just some really interesting developments that have come, and Carolina Gold continues to be at the center of stage at times, and so I'm excited to share this with you. So let's see if I know how to work this. This is our research center at Stuttgart, Arkansas, and our research is funded by the USDA, just like this facility here, USDA Agricultural Research Service. We're focused on a mission, and that mission is to try to do what we can to sustain and to help the U.S. rice industry. And so we're very focused on basic research, and we're at this center, it's just rice scientists, and our main area of emphasis is to explore the genetics, the natural genetic diversity that exists in rice. So one of the things that we're doing is we're curators of the U.S. Rice collection of that is here in the United States. And so we're responsible for growing out this entire collection. We don't do it every year, but we grow out a portion of it every year. And so this is just kind of an example of some of the diversity we would see in our field as part of this collection that's here in the United States. And so for years we've just been focused on characterizing this material for various different agronomic and grain quality traits. Well, over the last 10, 15 years, we're now being able to utilize genomic technology to actually look at the genes that are in these different varieties. And so this has opened up a whole new area of exploration and ways to explore natural genetic diversity in rice. So in our collection, we've got over 18,000 rice varieties from around the world. And the first one came and was put into the collection when the collection was first started, 1904. And so what this is just kind of a map of where some of these earliest earliest varieties of rice that were brought into the USDA collection, where they actually originated from. And the red colors are the ones where were the oldest varieties that were brought into our collection and where they were sourced from. And of course, rice is not indigenous to the United States. It's indigenous more to Southeast Asia. And so you can see where some of these first accessions came from and were brought into the United States for us to utilize and explore for potential production here in the U.S. And so then over the years, that collection of rice continued to grow, and so we have close to 19,000 accessions that we're responsible for. And so like I say, at our center, we grow out a couple of thousand of them every year to rejuvenate the seed, to characterize the varieties, to provide this information to researchers. And so if you looked at that collection of where all that material came from, this is kind of the diversity of countries that we have the collection from. And of course, we're fortunate that we can partner with the international collection that's at the Philippines, the International Rice Research Institute, where they have curate some 100,000 different rice varieties. So between them, we're able to share some materials and have access to rice genetic stocks that come from all over the world. So this is a pedigree chart that I've shown before, but it just kind of, in the United States here, we've had rice breeding efforts, so to speak, going on for about 100 years. And so the first rice that was brought into the United States were actually these, what we call these foundational varieties that were brought in from all these different countries from around the world. And in those days, of course, they would just try to grow those rice varieties, see if they were suitable. If not, they'd go and try and find something else. And so the growers, once they started identifying things that were successful, then it wasn't until, about the they would start identifying maybe variants within the field that were actually a little bit better, and so they would save that seed and replant that. Well, it wasn't until about the 1920s that they actually figured out how to start to make crosses, and from that point on, then they started making crosses amongst these lines and then starting to derive new rice varieties. And so we've essentially been doing that same thing for about 100 years now. And so one of the first varieties released as a result of those crosses was back in 1942, and it's just been a continuing process since then. And so one of the neat things to kind of look at this pedigree chart is that these lines up here essentially are the oldest ones with the newest ones at the bottom, and we can just kind of, by knowing how varieties are related to each other and what their parentage was, we know how these materials, what sort of predicted genes they might be carrying and where those genes might come from. So in our rice breeding efforts here in the United States, I was mentioning, so we started off with just bringing in materials from different countries around the world. And so there's been a study that has shown that most of the US rice varieties that are grown here actually trace their heritage back to only 23 ancestral plants or ancestral cultivars. And it was because only those 23 actually had traits that were useful for production here in the United States. And then it was amongst those that they started making these different crosses. And then through the breeding process, other selections were made. And so what happened is within the United States, the breeders found that they were able to really move forward the grain quality characteristics of US rice cultivars. And actually for a time, the United States was really considered the leader in the world in terms of rice grain quality. And the breeders essentially didn't want to bring any other diversity from outside because they'd essentially thrown away things that weren't working, and they'd refined this germplasm, this genetic materials, to come up with what they thought was really the best. And every time they kind of went outside of the box and started to try to bring in genetic diversity, what that nice little package of traits that they'd put together started falling apart. And so what we have is that in our US pedigree, there's very little diversity, and the breeders have just essentially been recombining materials within it. And whenever you tell them, well, why don't you go over here and get some of this really old material or try to another country, they go, nah, I'm not going to bother with that. And so during those early years of selection, what they were trying to do is get rid of these traits that are found in a lot of this world germplasm that don't work here. Lodging susceptibility, some of it was photoperiod sensitive, wouldn't even flower in our environments. And we also, as I was mentioning, really were pushing in terms of milling quality and of course yield and grain appearance. So we ended up with this very refined gene pool, so to speak, that the US rice industry relies upon today. But we've gotten to a situation now, of course, where we're looking at some pretty dire straits into the future. A lot of changes coming at us in terms of population growth. Are we going to be able to feed the world? Over half the world relies upon rice as a staple grain for them. We aren't producing more land. There's less land available, and what land is available is generally not going to be the best land. And so how are we going to feed the world into the future? And we're seeing, even here in the United States, a lot of stresses in terms of rice production, as David was mentioning, in terms of just water, access to water. At one time, we had 500,000 acres of rice being grown in Texas, and now it's down to about 130,000 acres, and that's being largely driven by lack of water because they've been going through a drought. Same sort of thing is happening right now in California, where they've been relying upon the snow melt in the mountains to irrigate their fields, and they haven't had enough snowfall for several years now. And of course, any industry can only last for a period of time without being able to sustain itself in terms of maximum production before it starts collapsing. So maybe it's time for us to start thinking about that genetic diversity again. And can we go back into that pot and actually find some traits that were going to help us kind of deal with some of these challenges that we're being faced with into the future. So when you have, like at Erie, 100,000 rice varieties, or in the United States here, 18,000 rice varieties, where do you start? How do you start looking at that material to try to make use of it? And so what's been going on is a lot of the research community has been developing what we call diversity panels. And these are like little core subsets of the material that's in these collections that is representative of the genetic diversity in that collection. And so you're working with maybe a subset of 200 or several hundred rice varieties, but that's much easier to work with than tens of thousands of rice varieties. And so there's been several of these what we call diversity panels developed. One is called the Rice Diversity Panel 1, which is one that we worked with Cornell University with Susan McCouch to develop. And we started off with not only coming up with this subset of material, but we've also started using the genomic technology to characterize it for genetic diversity at a DNA level. And so we started off with evaluating it with like 1,500 genetic markers, and the technology has really escalated, and so now we're looking at 700,000 genetic markers that have been scored on this diversity panel of over 400 accessions. And then Erie in the Philippines did something very similar. And they came up with a panel of 1,300 varieties. And they've also scored it with these 700 markers. And now there's a new group, you can't see it down here, a new panel of 3,000 genotypes from China that has been now scanned with over 2 million genetic markers. And so now we have this huge body of information of world genetic diversity that has been characterized not only for its physical traits, but also at a genotype or a genetic level. And so what we find is when we do this genetic analysis is that in this global diversity of rice, there's these relationships that keep showing up. And so there's two major groups of families. And so what this is called a genetic tree. And this is how these 400 different rice varieties are related to each other. Each one of these little lines relates to a genetic a variety. And so you have these two different groups. What's called up here is the Japonicas, and down here are the Indicas. And within the Japonica group, there's a temperate Japonica, which is the type of material you'll see in like Japan, northern Asia. There's the tropical Japonicas that are found in more tropical regions. And then there's the Indicas, which are found, of course, in Southeast Asia. And then there's another group called Aus, which is more in an area of northern India. And so now that we have these genetic markers, we can start seeing how these random rice varieties actually have some relationship to each other. And we can identify lines that are, say, here, just at a genetic level, are very different from these over here or at this end. And so if you're a breeder, you might want to say, I want to maximize genetic diversity in my next cross. Then this gives me ideas of how to make crosses for particular purposes. Now this was actually scored with the 44,000, excuse me, this is the one with just the 400 varieties with 36 DNA markers, just 36 markers. And so when we look at, I also wanted to mention that within this group of material, there's the tropical Japonicas, and that's where all of the US material comes from. And so all of the current varieties and the ones in the past essentially have come from this little neck of the woods here, and this is where the US breeders have their interests focused on. Now when you expand that tree and look at 3,000, we just were looking at 400 genotypes, and when we look at 3,000 different rice accessions, what happens is the tree looks pretty much the same. You've got the big indica set up here, you've got the Japonicas down here, and they're separated into the tropical Japonicas and the temperate Japonicas. And then we have what we call the Aus group here. And so what's meaningful of that is regardless of how many different genotypes that you look at, or how many different rice varieties, or how many different markers, there's these huge differentiations within this gene pool that makes something, makes a variety more similar to, or traces to its heritage of where it came from at a genetic level. So who cares? Well, of course, the breeders and the genetics community cares. And so what they're looking at is amongst these 5 different subpopulations of rice that we have, the indica, Aus, tropical Japonicas, and temperate Japonicas, and then the aromatics, that we've been able to identify how much genetic diversity is within those groups. And the indicas are the ones that have the most genetic diversity. So if you're trying to find something that's going to address climate change, people might say, well, that's where I want to go look, because my likelihood of me finding something is going to be greater there. However, the United States is really just interested in these tropical Japonicas, because that's where our history lies in terms of breeding. Let me just say the reason why I mention this is that most of the rest of the world is interested in looking at the indicas. And it's really only us in the United States that are really interested in the tropical Japonicas. And if we're interested in the point being that the world resources are not going to be looking in the corner of the world that us in the United States are most interested in. And so how can you use that kind of information? So this is an example. You don't need to see them here, but these are all different tropical Japonica rice varieties, many of them from the United States. And across the top here, we have essentially like the 12 chromosomes of rice just laid out end to end. So you've got chromosome 1 down to chromosome 12. And then along this would be just looking at the genetic diversity along that chromosome here for this first variety, which is M202. Should be able to find Caroline Golden here. And so what we see here, and this is looking at, these are all tropical Japonicas, and so when you see a red blip, the red blip means that there's a gene that's in that tropical Japonica background that's not tropical Japonica, but actually came from one of those other subpopulations. And in this case, it's coming from the indica. And so just as an example how this is meaningful, is here in chromosome 1 where this line is, this is where the semi-dwarf gene was introduced into rice. And so, as you know, Carolina Gold's about my height, and most rice varieties that are grown commercially are about more like waist height. And that's because the utilization of this one major gene, which has dramatically reduced the height of the plant. Why was that important? It allowed then growers to put on more fertilizer, which allowed the yields to be greatly increased without the rice being lost due to lodging. And so if we scan all this material with these genetic markers, what this shows us is, hey, right here is where that introgression or that gene has been put in. And so you can see the varieties that have it and those that do not have that gene. And then likewise, you can start looking at the rest of the genome and see where there's different sources of genes have come from. Down here on chromosome 12, there's another one here. This is a major blast resistance gene that has given us disease resistance. And so even if you don't know what you're looking for, you can say, well, I have a map here which shows me where these genes came from. And then I can start, when I can start looking at different rice varieties, I find out that they have traits of interest, I can start relating that to whether those are from a tropical Japonica origin or from someplace else. And just briefly how a breeder might look at this, here's an example of the variety Cypress, which was derived from a cross between Lamont and L202. And Lamont has great yield, great milling quality. L202 has great seedling vigor, and it's also improves the milling quality. And so what happened when the variety Cypress was developed, it became the gold standard for rice grain quality because it has just this beautiful grain and excellent milling quality. and it comes from a combination of these two parents. And so having this detailed genetic information allows us to essentially to scan these different chromosomes and find out, well, in this example, the green indicates that's where, this is looking at one chromosome, and you can just scan down this chromosome and find out where along the chromosome was the source coming from Lamont, and where was the chromosome was the genes coming from the L202. Now, as I mentioned, that the US material essentially traces back just to 23 different parents. And so what happens is that at a genetic level, that material, the material all looks pretty much the same. It's hard to differentiate it. And that's because there's so many of these areas of the genome that kind of trace back to the same source. And so if we did that kind of intense scan along these different chromosomes of rice, and this is looking at two different parents that were crossed, what we find are these regions in the chromosome that we're essentially blind to, meaning there's no genetic diversity there. Now, does that mean that there's just no genetic diversity, or is it because we can't see it? And so there's these, if you're trying to identify a gene controlling a trait, we know that there's certain areas of the genome that we're just blind to. And so part of the reason for that is all of this work in terms of the genomics of rice started back when they did a sequencing. They essentially identified all of the genetic bases that are found in a temperate Japonica variety called Nippon-bari. So that's called our backbone or our reference genome. Everything is related to it. Well, we know the temperate Japonicas are different than tropical. And the people that are interested in the indica material, they knew the same thing. And so they went and they sequenced their own variety, an indica variety called 9311. And so now they have their own reference genome, an indica, and the folks that are interested in the tropical temperate Japonicas have theirs. Well, as I mentioned, tropical Japonicas aren't grown that many places in the world, and so most people don't care. But we care. And so what we think is those desert regions that we find in the genome where we're just kind of blind, we think the problem is that we're trying to compare things against a backbone of genomic information that's just not meaningful. And so if we're trying to really explore the genetic space of US genetic materials, we need to have that genetic background that is relevant for our material. So what we've done in the last couple of months is we've taken all of this genetic information that's out there on the web. We've put together almost 4,600 scans of genetic material of 4,600 different rice varieties into one big pool of information. And this is essentially going to, it shows that same sort of tree that I was showing before, that most of the genetic material that people are looking at is indica, because that's where most of the rice is grown, is in those regions. That's where most of the genetic diversity is. And that what we see here, that there's a subset that's this tropical Japonica material that we're interested in, and then there's these temperate Japonica ones also. So now if we wanted to go in, and in this tropical Japonica set of materials, there's almost 800 different rice varieties. And if we wanted to say, well, which one's going to be our Nippon-bari? Which one's going to be our 9311 that we can choose as our backbone rice variety? Where do you start? How are you going to figure that out? So what we did is we took just that subset of material that's tropical Japonica, and this is what's called a relationship tree, and it's essentially using that genetic fingerprint of all those different 400 different varieties, and we identified how they're related to each other. And so we also wanted to do, what you do then is you come up with what's called a consensus genome. And what that is, they took all of the genetic information from those 400 tropical japonicas, and they found the genetic markers that were most frequently found across all of them. And so that's coming up with the markers that seem to be just generically common to tropical japonicas. And so we wanted to then use that genetic information, just like if you're seeing here, if this is a field of visually genetically different material, and we can, if we know that amongst these materials that they have a certain number of these genetic markers that they all have, that is essentially like saying that's where those genetic markers, or that's where those varieties all trace to. That's like their ancestral genome. And so, I'll go back here, whoops. So what we wanted to do is then find out that if this is a predicted ancestral genome, the genetic markers that are common to all this material, then which of our existing rice varieties might be most closely related to an ancestral tropical japonica. And so what we did is we evaluated a number of these different varieties of how similar they are to this ancestral genome. And this is what's called a distance matrix. And so the varieties that are up here are the most closely related to the ancestral genome, with the ones as they go down further are further related. And so in this set of 400 different accessions, we had a number of different Carolina gold accessions that were in there. And what we find is that the Carolina gold rice is very similar to this ancestral tropical japonica genome. And so if we go back and then say, well, of these tropical japonica accessions that are closest to these ancestral tropical japonica genome, where did those come from? Well, we know that we claim that the Carolina gold one comes from the United States because we don't really know where it came from. But if you look at the dots that are the red ones, or the red or the yellow, those are the ones that are the most ancestral-like, the oldest. And so you kind of start seeing where they came from. And some of these kind of tend to come out, you know, related to maybe with the slave traffic at one point. There's some over here in India. And so this kind of says, well, where would those genes originally have come from? What parts of the world? And so what we're interested in doing is, because we know that Carolina gold was brought into this country, was one of the first varieties that was really commercially utilized, we're able to go back into this pedigree map of US materials and trace, just by pedigree by descent, where would, this is Carolina gold right here, where did it have its influence in terms of current day US rice varieties? And you can see that Carolina gold has been instrumental in having its influence on current day rice varieties. And so not only has it been instrumental in how breeders have essentially been reutilizing and recycling those Carolina gold genes, we also know that Carolina gold is very similar to an ancient tropical japonica. And so when we come down to say, which rice variety would we want to use as our background, as our reference genome, we can make a really good case that Carolina gold would be an excellent variety for that. So how would we use that type of information? Well, what we want to do is take Carolina gold and conduct a complete DNA sequencing of it. And that means we're going to know all of the genetic information it possesses. And with that information, that's going to be of benefit to the US rice breeding programs, because we're going to have a more accurate DNA baseline from which to compare materials to. We're going to have a finer resolution to actually be able to genetically differentiate our current rice varieties. And as I mentioned, we have these huge gaps that we don't even know if there's genes that occur in those gaps. We're going to be able to fill in those gaps. And so ultimately, what we want to be able to do is actually be able to relate the actual DNA structure with specific traits that are found in U.S. rice. And that's going to then help breeders make new combinations of genes for developing superior rice varieties. And so you might think of it in terms of, right now, we have a rough genetic map that's being based upon these temperate Japonica and Indica genetic background material or reference material. And it's somewhat like you might think of the interstate system as a map within the United States. But if you want to get off the interstate and go find a little town, you're going to need some more information. And so likewise, once we get more information on Carolina gold as our reference genome, we're going to be able to explore our own tropical Japonica germplasm much more effectively. How might this be of interest to other folks like the Carolina Gold Foundation? Well, for one thing, there would be researcher community around the world as well as research resources focusing on Carolina gold, because it would become a new reference or a new standard for the genomics industry, rice community. We also may find that Carolina gold has a unique genetic signature. And so with using that type of information, we can then go find out, well, where did Carolina go in the world? We have some maybe anecdotal evidence based upon names. You find varieties in South America that have Carolina as part of their name. But we'd actually be able to do this at a genetic method, not just by name. The other thing is, where did Carolina gold come from? We've never really known that. And so we could use this same sort of technology to be able to compare Carolina gold today with other genetic resources from around the world and find out, does that fingerprint exist elsewhere? And so I'm in the midst of people that know a lot more about Carolina gold's history than I do. And so I know there's like the Carolina sisters, the gold and the white. You know, how are they different? Why are they different? Why are they sustained? And so there's a number of different things that one can do to learn more about the culture and the history of Carolina gold, as well as this having a positive and very significant effect in terms of how this information can be used for further breeding efforts in the United States. So with this, I wanted to introduce you to the other group of people that I work with at our center, and particularly Jeremy Edwards and Angela Baldo, who has just recently joined our team, and they are the bioinformatics people that have been utilizing this information and helping us to kind of tease out new hypotheses that we can now ask with this genetic information. And with that, just wanted to present to you, this is kind of a hypothesis that we're going to forward with our research community, that this should be the variety that we choose as our new reference genome for tropical Japonica germplasm. So thank you, I appreciate your attention.
This is really exciting work, you know, with the genetic resources and advancements in sequencing technologies. Are there going to be more reference genomes sequenced in rice in addition to Carolina gold with the community around the world now that the prices are so low and there's so many people interested in the technology?
So it has been proposed that, you know, eventually they're looking to essentially at least resequence all of the Erie collection at some point. That's 100,000 rice cultivars. Of course, I think the sequencing technology is going to have to get much less expensive, but that is their goal, and of course that at some point, that probably will become a reality at some point, but it's going to require significant financial support to make that happen. So yes, that the sequencing technology has dramatically dropped, and it just seems to be, the cost of it has dropped. And that, every 5 years is just a dramatic change, and so you're able to do a lot more with a lot less resources. So yeah, that day is coming.
Anna, thank you. This is really exciting. The idea, is it ridiculous? Well, maybe it is, to talk about drought-tolerant rice as we move into water conservation. And is there anything to be said about the genome work and how it can apply to moving rice cultivars into the moist ground arena where we're conserving water and staying aerobic more so we avoid toxic metal uptake versus how that plays out in future rices?
Yeah, I actually think that's probably the most compelling area of research is just, I mean, we've seen it here in the United States, the loss of rice acreage driven by the lack of water. And so, and we're not the only place in the world that's having that problem. And so we have to change rice production to use less water. There are rices from around the world, you know, places like in South America and Africa, where it is grown in what we call an upland situation. It's not flooded. They're just essentially relying upon rain. And so we know that, but those rice varieties, according to our standards, are not high yielding. But they're able to sustain, you know, living in those kind of conditions. So we know that those genes are there. And like I say, you know, breeders have been hesitant to try to work with some of these materials because you might bring in drought tolerance, but you might bring in other things that you didn't want. And so I think this really gives us the opportunity to really explore what materials have the traits that we want and how you can surgically essentially move in just the traits you want and leave behind those traits that are associated with not being adapted to our environment. So there are many places in the world that are starting to look at rice essentially being produced without a flooded culture because they too are going through that same situation. I think there's a second part to your question. Is it?
The second part of the question.
Sorry. Short-term memory.
The second part of the question was in when you're moving obviously into moist ground versus flood environment, you're moving away from anaerobic culture, the chance for anaerobic culture into aerobic culture. And does this help clarify when you're talking about surgical identification and movement of segments of the genome? Does this help obviously clarify how toxicity and uptake would work in rice systems and possibly how to maximize aerobic culture while minimizing other uptake in an aerobic system, which gets really confusing?
So I think you're primarily focusing on like the arsenic aspect.
That and the other heavy metals too.
Right, okay. So yeah, that gets very complex, and so we know that just naturally occurring arsenic in the soil or cadmium in the soil is, you know, can be taken up by the rice plant. And so it's interesting because it's not just the genetics of the plant that are going to be important here. It turns out it's also going to be the genetics of the microbial community in the field, which shifts dependent upon how much waterlogged it is, how aerated it is, what the soils are. And so it really goes into a whole nother level of complexity. You know, I've been talking really about genetic markers here, but there's also another area where you look at like expression of genes. What genes are actually turned on or turned off in different environments. And so I think that's really going to be another area of how do you find rice varieties that have the right genes turned on or turned off to prevent some of these events happening in terms of metal accumulation or to prevent susceptibility to diseases under drought stress conditions. So there's going to be a whole new level of exploration for breeding purposes that will be, it has to be tied back to the presence or absence of these genes that we know through these genetic markers.
Anna, in the 1840s, there was a spontaneous mutation that took place in Carolina gold, creating a long grain version of Carolina gold. This rice lasted until the Civil War, when circumstances caused it to become extinct. It was this long grain version which commanded the highest price in the world grain market, so we've naturally always been interested in it. It sounds, though, that if we had a complete genetic map of Carolina gold, we could conceivably locate those places in the gene map and the alleles which govern the size of grain, and they could be directly tweaked. And we wouldn't necessarily need to have the genetic material from a surviving 1850 sample of grain in order to do that?
Absolutely. And whether that would be done through, more than likely it would be done through just breeding, incorporating those genes that control grain length. But like you were saying, you could do this kind of surgically, not disturb the rest of the background, but just those areas that you wanted to disturb.
Well, I see a really tremendous benefit, of a practical sort arising from this map, just for the variety that.
What was that called, that variety?
It was just called long gold.
Okay.
Okay.
A new long gold.
Well, thank you so much.
Thank you. Thank you very much.